question number 4 up here so that I can walk through it with you. But we did the same process for questions 1, 2, and 3. But we just have the numbers written down for that. So, yeah, to get started, the question says that a cross is done between corn plants. One's heterozygous, the other one's homozygous recessive. The recessive genes are anther, ear, brachytic, and fine. And so we showed our test cross right here. The one is completely heterozygous, crossed with the one that's homozygous recessive. And then we found the 2 that were the most abundant. We realized that those were the phenotypes of the chromosomes that are on the heterozygous parent. So the gene arrangement is wild type, brachytic, and fine on one chromosome, and on the other one it's anther, wild type, and wild type. And then to find the gene order, we looked at the least frequent, which would be the double crosses, and we found that the gene order is wild type, fine, brachytic, and then so that, well, I guess that ends up being anther, fine, and brachytic in that order. And then to find how many map units away the anther and the fine genes were, we added up the 2 double crosses and then the other crosses that were just the single crosses between the 2 first genes. And that ended up being, there were 190 offspring that had that. Over the 1,000 total offspring times 100 gave us 19 map units. And then to find how many map units away the fine and the brachytic were, we just took the double crosses first, and then we did the crosses where we just flipped the third one, and that ended up being 20 and 26. So the total was 51 over 1,000 times 100 gives us 5.1 map units away. And then to find the interference, we did 5 double crosses over the possible double crosses, which was 9.69. We found that by the frequency of these or the distance between these and their decimal units. So it's 0.19 times 0.051 times 1,000 gave us 9.69. 5 over that gives you 0.52. 1 minus 0.52 gives you 0.48. So that was the interference. And so that was how we did question 4. We did the same process for question 1, and these are the answers we got for that. We got A, wild type MG for the allele arrangement for one of the chromosomes, and the other one is the wild type R wild type. And then for B, we got the order is the R, the A, and then the MG. And then for C, the map was that R and A are 15.01 map units away, and then A and MG are 15.5 map units away. And the interference we found to be 0.577.
So then for number 2, what we found was for the gene arrangement, it was a V with the SC plus, and then S, and then V plus, SC, and then S plus. And this was also the order, V, SC, and then S. And then for the linkage map, right, we found between these 2 was 10 MU. And then between these 2 was 33 MU. And then our interference value, if finding everything the same way, was 27% or 0.27.
All right, and then for number 3?
Yeah.
Okay, and then so the numbers for number 3 are for part A, it is wild type C with GS wild type. And then the order is also in that order, which is GSC. And then the linkage map is 1.9 map units between G and S, and then 5.6 map units between S and C. And our interference value for that one was 1.
Make sure I got that one right. 3D. No, I think I did that one wrong. What's 3D? What do you have for 3D? 3D. Yeah.
Oh, maybe it was.
interference number?
No, that's what I got.
Oh, that's
Yeah.
what you got. Okay.
Yeah.
It is one.
Yeah.
It is one.
So yeah. Our
Oh, right.
interference value was 1 for that one.
Yeah,
because there were no double crosses
Exactly.
in
Okay.
there. So
Okay, and then for question 5, just trying to figure out or compare independent assortment to linkage. For A, we got a 1-to-1-to-1-to-1 ratio of big A, big B, to big A, little b, to little a, big B, to little a, little b, and each one is like 25% likelihood. And then B, we got 50, like 0.5 to 0.5, big A, big B, and little b, because they can't cross. So you can only get, like if you get big A, you're going to get big B. If you get little a, you're going to get little b. And then for C,
Just like A.
we figured it's just like A because there's a likelihood that you can have the combination of the alleles, even though big A and big B are linked. Since there's crossing over, you can get one of the sister chromatids is going to be crossed, but the other is not. So the other one is still going to have the big A, big B, and the little a, little b. And so that was a 1-to-1-to-1-to-1 ratio because you didn't give us how close they were. And so for D, when you have to deal with how many map units away they are, we figured that it's 0.078 chance that you're going to have a cross. And so that means that, is it 90, how many, what is that, 90 something? Let's
Nice.
100 minus 7.8. 92.2% of the time, you're not going to have a cross. And so like the frequency is just half of that for big A, big B, and little a, little b. So we said for big A, big B, 46.1, and for little a, little b, 46.1. And then for big A, little b, and little a, big B, we had half of 7.8, so it's 3.9 and 3.9. And that's it.